Okay, it looks like it is time to begin. So I'd like to welcome you all to the United Leukodystrophy Foundation's virtual family conference session on Global Leukodystrophy Initiative, presented by Dr. Vandiver, Dr. Eichler, Dr. Fatimi. This session is being recorded and will be posted for viewing online in early July. Thank you so much for joining us. And now I'll turn it over to Omar.
All right, thank you very much for the introduction, Tara. So thank you all for joining us today. We are the Global Leukodystrophy Initiative Clinical Trials Network, and today we'd like to just give an introduction to our consortium, talk a little bit about what we do, and then especially about how you can support this effort and how we hope to give back to the community. So maybe I'll introduce myself first. I am the research program manager for the Glia CTN, and I will also be introducing my colleagues, the principal investigators of the Glia CTN, Dr. Florian Eichler, Dr. Ali Fatimi, and Dr. Adeline Vandiver. So the agenda, let me just move this forward. The agenda for today's talk is pretty straightforward. We'll start with an introduction, just so that everybody knows, who we are, what we do. Go through our projects, the research projects that we're currently working on. Go through some of our engagement models. Talk about how we work with the community, especially with our advocacy stakeholders. Talk about how we support educational efforts and career development for other physicians who are interested in getting involved in leukodystrophy research or clinical care, and then really get into how the community can participate and what you can do to support this research, and then finally leave some time for questions, and really kind of try and get at what we can do to support you. So with that, I'll begin with my introduction. So basically, who are we? So just some numbers, some very basic numbers to kind of put everything into context. So we are a research consortium that consists of 8 clinical centers around the United States. I'll show a map on the next slide, so you can all kind of get an idea. And we basically do research, different types of research, but our observational study has enrolled over 2,200 participants to date. This is a project that started many years ago and is now being supported by a research grant. So we're building on a really large amount of legacy data and hoping to use that data in new ways, and really be able to use it to its maximum potential. As I mentioned before, we work very closely with the advocacy community. We currently have 40 advocacy partners, over 40 advocacy partners, I would say, who are involved in our committees and participate very actively in the consortium. And as I mentioned before, we are currently being funded by a federal grant from the National Institutes of Health. It's a 5-year grant that is renewable for up to 3 cycles, so for potentially up to 15 years. And I'll just read through our mission statement very quickly, just so that everybody has an idea of really what we're trying to do. So we are a consortium of institutions, clinicians, scientists, and patient advocacy leaders working together to promote advances in the diagnosis and treatment of leukodystrophies. Specifically, we seek to create a robust research infrastructure that will allow for collection, sharing, and analysis of standardized longitudinal natural history data, development of novel clinical outcome assessments, and identification of surrogate biomarkers, ultimately paving the way for transformative therapeutic trials across the leukodystrophies. In parallel to these approaches, we work closely with a diverse group of stakeholders to promote disease awareness and education, advocate for the adoption of universal newborn screening and early diagnostic programs, and establish clinical guidelines to support the short and long-term care of individuals living with leukodystrophies. So Our participating study sites, this is a visual. As you can see, we are fairly well distributed around the country, and that's that's not by accident. We want to make it easy for individuals to engage with us regardless of where they live. And so that is why we selected these sites. More information about these sites is available on our website, and we'll post a link to that later on. But I'd also like to emphasize that we do work very closely with with other researchers and physicians around the world. These are individuals who are not currently funded by by the research grant that I mentioned earlier before, but who do contribute in very meaningful ways to the research that we do, and who have been very valuable collaborators for many years. And this is also just a partial list. These are the individuals that we're working with right now on active research projects, but we've worked with many other leukodystrophy researchers in the past. So I'd like to introduce some of the key stakeholders within our consortium. So as I mentioned before, our executive committee is run by Dr. Vandiver, Dr. Eichler, and Dr. Fatimi, all three of whom are here today. And this is the executive committee that really is responsible for, for overseeing the consortium, making sure that our scientific efforts are going well, that funding is being used appropriately, and for providing progress reports and coordinating administrative duties. So beyond that, we are also represented by the advocacy community. So we have an advocacy co-chair and a chair. Maria Kafalas has been the chair during this first year, and Erica Barnes has been our co-chair. And essentially, the advocacy committee is responsible for interacting with other committees. You see these listed on the slide, and really just providing oversight to make sure that the activities that we're doing through this consortium are really aligned with the priorities of the patient community, and that we don't lose sight of that. And of course, the advocacy committee also meets internally, really to align themselves around priorities and communicate those to us. As I mentioned before, we do work with a very large number of advocacy groups. And so it really is a very important job that Maria and Erica have done, to kind of keep everybody informed and keep everybody on the same page. So other committees, we're limited on time, so I won't go through this in too much detail, but you'll be able to look at this slide in the recording. I'd like to emphasize, mainly the data governance committee, which is responsible really for making sure that that the data that we collect through this consortium can be shared easily and appropriately with other stakeholders, and that includes other academic researchers, the advocacy community, our industry partners. And basically what I'd like to underscore is that it is really essential to us, and really a very high priority for us, that the data that we collect, due to the contributions of the community, are then made available to the community. And of course, that we obviously understand that there are safeguards just to make sure this is sensitive data, and so obviously we take appropriate steps to protect it. But at the end of the day, we really want this data to benefit the community and help us all, get towards the goals that we're working towards. And the other committees we'll discuss in brief throughout the presentation. But I will move on and hand it over to Dr. Vandiver.
All right, thank you, Omar, for that introduction, and thank you everyone for your attention. This is a project that, as Omar says, has been sort of in the works for many years, and Bob Rauner of the ULF, as well as other advocacy partners at the MLD Foundation and at the PMD Foundation in particular, along with Hunter's Hope and Mission Massimo all were part of an effort that started as far back as 2013 to really focus our efforts as a joint community and as a partnership to achieve clinical trial readiness in these disorders. And it's been really gratifying to see a lot of the advances across the field, not just from our consortium, but across the field as we work together on these collaborative approaches. Next slide. So it's my goal to explain a little bit to you how we've structured some of the research projects within the Glia CTN. And it's important to keep in mind that the kind of work that we do as clinicians and scientists is obviously only part of everything that needs to happen within the disease community, but that we sort of need to translate some of those loftier goals into really discrete and sort of granular metrics so that we can show our partners at the NIH and other people who fund us sort of return on investment and sort of moving our community along as far as clinical trial readiness. And so we were allowed to do a number of projects within this research grant, and we focused on 4 projects, and you're going to see that we focused on some specific diseases. And I want to emphasize that the focus around those diseases was thought out not just because they were, for some reason, favorite diseases, but more because in many ways they represented the needs of the disease community. And one way they represented the needs of the disease community is that these were some of the disorders that were the closest to therapeutics at the time when we planned these projects. The other way that they represented the needs of the disease community was that they were actually exemplars for other leukodystrophies. So for example, adrenomyeloneuropathy with a lot of problems that come from spinal cord involvement, the tools that we can develop for that disease will be applicable to some of our other adult onset leukodystrophies. Alexander disease with its tools and its focus on some very specific what's called biomarkers, which is tissue sampling, will help us prepare for other leukodystrophies and for the therapies that might be available to other leukodystrophies. And then Aicardi-Goutieres syndrome is a very early onset disorder. And so focusing on some of the most severely affected of our patients will help us to understand how to measure outcomes in the sickest of our members of our community and how to support them. And then overall, in the first project, we focus on a broad number of different disorders, really building infrastructure and systems by which we can have a toolkit that's applicable to a broad number of different disorders that are within our leukodystrophy community. Next slide. So the very first project is something called a natural history study. So a natural history study is something that allows us to understand what you expect the course of an individual disorder to be and what you might expect the course of an individual patient might be if they have no therapy and no intervention in sort of its quote unquote natural state with just the disease evolving as a disease might evolve. And that information is very important because it allows drug companies and our scientific partners to understand how they might be able to plan a clinical trial that might be able to change how that disease progresses over time. And also because it might allow us to understand the impact of an effective therapy and make justifications towards governments and insurance companies about the need for implementing treatments, even if they might be difficult, potentially invasive and expensive. So the way we're working on that together as a community is that we have a total of 8 sites that are distributed not fully equally, but the best of our ability across the United States. And I want to emphasize for those of you who are international partners that it's not that we don't collaborate and don't wish to collaborate with our international partners. And as a matter of fact, we have partner sites for some specific disorders across many of our global partners, but more just because this particular grant required participation from US sites because it's funded by the US it's funded by the US government, right? So these are US-based sites distributed across the United States, all of which already saw and took care of large numbers of children with leukodystrophies, and so would be expected to be able to handle additional patients who might come in for research assessments. Next slide. Then the next project, Project 2, dials down more specifically into a disorder called adrenomyeloneuropathy. Now, as part of Project 1, right, we also look at patients with ALD and AMN, and we have them do a series of, along with the other patients that are covered in that Project 1, a series of standardized functional assessments, sort of what is currently the gold standard in clinical trial research around measuring how well people walk, or how well they are able to move, and or how well they're able to communicate, right? We also measure how something called patient-reported outcomes, how they feel about how they do. But in this project, Doctors Florian Eichler and Doctors Fatimi, along with their collaborators at their institutions, are working specifically to develop novel tools. And some of those novel tools, right, especially during such difficult times of the pandemic, are expected to make clinical trials a lot easier and quantifiable for patients with this type of disorder. And specifically, right, they are piloting novel wearable technologies with instruments that you can wear on your wrists and on your ankles to sort of understand how we might be able to capture, right, how people are doing and functioning without having them need to come to a specialized medical center to understand that information. So sort of in the long term, the ideal would be that you travel to a specialized medical center to talk about a potential therapy. You potentially have that therapy started, right? But then rather than having to come back every couple of months to that doctor and make what might be a long and difficult trip, especially if you either live far away or your level of disability is very high, rather than having to make that difficult trip back on a very regular basis, you might be able to actually have some of those metrics that were previously captured by specialized physical therapists and specialized equipment in a lab setting or in a hospital setting, be able to actually have that captured by equipment that can be shipped to your home. And so Dr. Fatimi with Dr. Fine and Dr. Bastian, and then also Dr. Florian with Dr. Lewis and Dr. Stephen are really working on these novel approaches specific to AMN in this grant, but that might be able to be applied to lots of other leukodystrophies to help us understand how we might be able to make it easier to understand how a disease progresses and how we can capture that information while limiting the burden of travel and participation in clinical trials for families. Next slide. And then in Project 3, right, we are working on a disorder called Alexander disease, which just had the onset of a new clinical trial, right, that looks at the ability of a special medication called antisense oligonucleotides to decrease the production of a protein that's toxic in Alexander disease. And as part of this project, we're showing how effective, right, the network is at being able to collect data on GFAP, ship samples, and collect information, as well as showing what the relevance of that biomarker is, specifically, for example, if you have a higher or a lower amount of GFAP, right, in the samples that are taken from an individual patient, is it likely that that corresponds to a higher or lower level of motor function or other type of disease progression, so that as treatments are being developed for this disorder and being trialed, we can robustly say that if you were able to show that you could decrease the GFAP, that would mean you could also expect that you were decreasing the burden of that disease on an individual patient. And that may sound very esoteric, but where it becomes important for clinical trials is it might take years to show for an individual patient that a patient's motor skills or other impact from their neurologic disease is better, or worse in the face of a treatment. But if we know that once you can change the GFAP, you can expect that in the future, the person's motor skills or other disease-associated manifestations are improved, then you could take shortcuts in clinical trials and make them last less long and have answers more quickly. And that showing that we can do that and get that kind of information is important, obviously, to Alexander disease, where there's ongoing clinical trials, but might also and would also be important to other disorders, where if we show that we can successfully do this and potentially then also use these tools to do this again in other disorders, show that that we can meaningfully that by meaningfully changing a protein that's overexpressed in a particular leukodystrophy, which Alexander disease is not the only one that's in that category, we can also show that we would expect in the long term that we can change the disease course. Next slide. And then the last project, right, is looking at developing novel outcome tools. So outcome tools, right, are tools that we can use to quantifiably measure how a person is functioning who's affected by a specific disease. And most functional outcome tools in the leukodystrophies were actually developed for other conditions. ALD may be the exception to that because it's been known for such a long time and been studied so well and so robustly. And there are a couple other disorders like metachromatic leukodystrophy that have disease-specific outcome tools. But in most of the other leukodystrophies, if you're trying to do a clinical trial, you're actually going to be borrowing, right, an outcome tool from something like cerebral palsy or from another disease that may or may not have a lot of overlap with a leukodystrophy. And so it's important for our disease community to start working together to develop outcome tools that are made specifically for these disorders, because it's the most likely that those tools will be the most helpful to measure a change during a clinical trial. And so Project 4 is focused on the development of a specific outcome tool for one of the more severe leukodystrophy, Aicardi-Goutieres syndrome. And in those leukodystrophies where children are very severely affected, and again, AGS is not the only one, it can be very hard to use an outcome tool that was developed for another disease like cerebral palsy in our affected patients, because the patients are sicker and have worse motor function than most children with those other disorders that the tools were developed for. And so as a community, being able to work together to develop disease-specific outcome tools becomes very important. And here's where the participation and the collaboration with our patients and our advocacy groups becomes particularly important, because it's important for us to hear from the families that we're measuring the right things and things that are important to our families. Next slide. So in parallel, right, with these efforts around working on tools and a toolkit for specific leukodystrophies that might be translatable to other leukodystrophies, our group is also working to save samples and save information about a broad range of different leukodystrophies for the future. And the word biobanking is really apropos because it's like putting money in the bank, right? It will bear fruit later. So, you know, when you go to one of our clinical sites or another doctor's office and participate in a natural history study and a sample is taken, that sample is typically saved and put in a bank to be used later when there are specific questions about how a disease progresses, and if there are specific things that you can measure in that sample that might show how the disease progresses or create a quantifiable metric for how the disease progresses, or also to be used later to understand how we can measure the impact of clinical trials. Next slide. So we've been collecting samples, right, in my lab well before this project started, and it's been almost 20 years now. And over time, right, that all that data information was moved to the Children's Hospital of Philadelphia, and then also provide storage for all sites that are participating in the Glia CTN. And the goals of that sample in the beginning were really to be able to use those samples to understand the genetics of leukodystrophies, and people who contributed to that biobank early on helped us contribute to and build genetic understanding of our family of disorders and to help us identify new genes, right? But in the meantime, also now with novel technologies allows us sometimes to take cells from those samples and to build models that allow us to test new drugs, as well as the biomarker discovery and evaluation that I mentioned before. And these samples are important to our community because there are very few people who save large-scale numbers of samples on this disease patients, and we frequently have requests for sharing those samples and have methodologies for sharing those samples so that we can facilitate the research done by other groups as well. Next slide. And this is just an example of the kinds of samples that we might take, right? Most commonly, right, it's a blood collection, right? And as simple as that, and the blood is saved, to both keep the cells that are in the blood, as well as to keep the liquid that surrounds the blood cells, which is called plasma or serum, depending on how it's collected, right? Sometimes also, if patients are having specific procedures done, we'll keep something called a skin biopsy, where we can grow cells in the lab and keep them for special experiments. If patients are having other tests as part of their diagnostic testing, such as a lumbar puncture, we'll ask to save samples. And then sometimes patients are going to have samples taken for other purposes, right? Such as a biopsy of muscle and things like that. And if those are available, we don't do these assessments just for the purposes of research, but if they're being collected anyways, we make sure that every tissue is saved so that nothing is wasted, and that there's extra tissue left over from the kinds of tests your doctors might do, that we save it for future research as well. Next slide, please. And all of that, all of those types of samples, right, are used as the precious resource they are. So cells might be grown in the lab and then frozen and saved in the long term for future biomarker studies, and to see if we can treat those cells to show that we can make cells better and have the hypothesis, therefore, that we can make people better. We do different sequencing approaches on the tissue, including DNA and RNA sequencing, and we can also look at tissue and cells to better understand how the diseases cause the symptoms they cause. And it's important to know that all these samples are, saved in very specialized freezers with lots of systems to protect them, both from the perspective of the safety of the samples, and the sample integrity, but also from the perspective of keeping the identity of those samples coded and protected, so that the confidentiality of the individual person who's shared that sample is also protected for the future. Next slide. Okay, go ahead, next slide. So I think oftentimes it's difficult to sort of explain what exactly a natural history study is and how a natural history study plays a role in helping us understand how to be ready for clinical trials and how to be ready for new drugs, right? And I think it's opportune to mention that both the Food and Drug Administration in the United States and the European Medicines Agency in the EMA and corresponding agencies in other countries that often look to those two large agencies for how to prepare for clinical trials, right, require at this point that we share information about how the treatments that we're proposing change outcomes that are meaningful to patients. And so without an understanding of the natural history, it's not possible to plan a clinical trial and to know, for example, how many people you might need to test a drug on in order to effectively show that the drug works, right? Impossible to know what type of change you want to measure to show that a drug works, right? And how to plan for a successful clinical trial. But natural history is also important to help families and patients know what to expect. When I see a patient with a new diagnosis and I tell them that they have that disorder, Almost 100% of the time, the very first question out of the family's mouth is, well, what does this mean for my child? How well will my child be able to eat? How well will my child be able to walk? How well will my child be able to talk? What other health complications might my child, or, and I'm saying child here, but it also can be an adult and a loved one, or for yourself, right? The only way to get at that information is to do natural history studies so that we can better predict what complications might occur, what needs to be monitored, and when key medical events are likely to happen. Next slide. And I think it's also important to talk a little bit about the difference, because I think this is something that's confusing, as our advocacy groups are also working with us on data collection and also working on us in ways to collaborate around preparing for clinical trials. It's important to know that a natural history study is actually often different, although there's some overlap, from a patient registry. Most often, registries are going to collect information on numbers and types of patients and how things might affect those patients at a given time, right? And might, in some cases, for advocacy groups that are working hard on this, look at what happens to patients who have newborn screening follow-up. But the type of studies done in research settings as natural history studies are a little bit different, and collect often more longitudinal information, and information in a more less open-ended and more pre-planned way about how things evolve in a disease over time, and also collect samples in a way and some clinical data in a way that's usually not permitted in patient registries. Next slide. All right, so here is some information about the patients that we've enrolled since this project has started in the past couple years. And you can see that a number of disorders have already enrolled substantial different patients, that there are also good numbers of patients with even some of the more rare leukodystrophies, even if the names are not up there, right? But that is in particular for disorders like Aicardi-Gouttières syndrome, Alexander disease, metachromatic leukodystrophy, TUB4A related leukodystrophies, and PMD, that our group has made substantial inroads in collecting and analyzing information about what for rare diseases are fairly large numbers. Next slide. We also want to share that the patients in our registry are distributed broadly across the country, right? And we want to work to support also the collection of data on states that might be less populated and less well represented, but that we really work to facilitate enrollment for families all over the country. Next slide. And even, although most of our enrollments occur from the United States, even all over the world, and that our team is poised and prepared to help families that are enrolling from other countries as well. All right. The next part, how do we work together with multi-stakeholder engagement? Next slide. So this is a very important piece to us, because the reason why we do this is not obviously for this data to stay stuck sort of within the walls of an academic institution, but really to work with other groups to share the information. So first of all, the Glia CTN, right, works to disseminate information as much as they can, right? They publish information. We are making as available as we can. It's some work in progress, but information about data and sample repositories, so outside groups that are not part of the Glia CTN know that the data is available. We work closely with our partners at the NIH, and they're stewards of data, something called the DMCC, to share data with them so that there's a long-term place for the data to live, right? We work to support smaller academic projects within the Glia CTN sites, and a number of our investigators are already taking advantage of that, right? And we also work with industry partners to support clinical trial development. But one of the areas where we would most like to see additional interactions and engagement is with our patient advocacy leaders. As Omar mentioned, we have over 40 patient advocacy leaders within the Luca dystrophy community, which is a large and wonderful number. And in many cases, those advocacy partners are already very well established and very successful and have a large infrastructure of their own. When that's the case, we'd love to and are working on making connections so that the type of data we can collect that's a little bit different than the type of data that those advocacy partners are able to collect better informs the important work that those advocacy partners are already doing, such as advocating for a newborn screening policy, interacting with the FDA, which requires them to bring sort of more information to the FDA about what the disease is like, participating and collaborating with industry partners, and preparing for clinical trials and referring to clinical trials. As much as the information we collect can help support those existing advocacy leaders who already do that type of work, we would like that to happen more and as often as possible. In addition, right, we want to make sure that those advocacy groups that are younger or might not have the same kind of infrastructure sort of get a leg up with additional infrastructure that we can provide so that they don't have to reinvent the wheel. And in particular, one of our partners through the NIH, the DMCC, can offer support to advocacy groups to create patient registries for individual diseases. You can imagine that type of close partnership is around information that's collected from patients is going to be regulated by ethics boards and sort of careful management of that data. And so we're working with our patient advocacy groups, and in particular with the chairs of the Patient Advocacy Committee, to really formalize how to do that. And we're also working with larger groups like the ULF that has a specific task force around some of this type of data collection and stewardship to find the best way with the right permissions to create long-term and longitudinal approaches to build the data everybody needs to do the important work they do. Next slide. And here are members of our advocacy community. And I think that this goes over several pages, so I'll let alphabetically, but we can go slowly enough so that if any of you that are on this call don't see your foundation or your disease foundation on here, please note that this is not a sort of members only club, that we're happy to have as many partners as we can, because we really want to create a community that works together and learns from one another and creates a lasting partnership for how to do this work as effectively and efficiently as possible. All right, and with that, I'm going to hand over the virtual microphone to Florian Eichler, who's one of our co-leads with Ali Fatimi, and who leads specifically the career development and pilot project efforts as such an important effort to build the next generation of investigators.
Thanks, Adeline. That was a very nice overview. And just to emphasize that a large part of this effort that we're trying to achieve across multiple sites and stakeholders is towards clinical trial readiness for these rare diseases, diseases that for many decades have not seen any treatments arrive, and where we also we don't understand the outcomes and often the longitudinal course well enough to design trials. But I think another important part is that we are really lacking in site investigators and trialists for many of these trials that we are planning, and we need to support and foster the next generation of investigators. And part of what we're doing here is to try to enable career development for these new young investigators that can then help lead future efforts and future trials. So I think it's an important investment in the future. Omar, next slide. So to briefly outline that we are aiming to fund one pilot project per year. And these research projects are those that should address key knowledge gaps that might not be funded without our help. Similar to NIH grants and overall important research proposals, these are grants that should be innovative. They should address an important gap and also help support the career of a junior investigator. The successful project should be related to specific established leukodystrophies, bring together really the biological problems with the unmet need, so that we're really aligning some of the focus around these single gene disorders, be aligned with what the patient community needs, what the advocacy groups identify as important gaps, and they could really range in their subject matter from biomarkers to clinical outcome measures. In general, what we're aiming for here is clinical trial readiness. So this is generally not work that is supporting preclinical non-human models. So we think there are other alternative ways to support preclinical testing in animal models. This is not to say that we are not supporting basic science efforts, but it should be connected to the human condition and really help therapeutic translation to the bedside. So it's really a very focused goal within these pilot projects. And we think that it really has the potential to serve two different goals. On the one hand, foster the career of a promising young investigator, and on the other hand, really address important unmet need in one of the many rare diseases that we're focusing on. Next slide. So I think a very nice example of a pilot project award was put forward by Eric Malik from Cornell, and he is focused on a prospective natural history study in boys with early adrenoleukodystrophy identified by newborn screening. And this has really been paving the way to hopefully scale up assessments and standardization across centers in the future. There are really two important aspects of this early monitoring and screening in ALD. It's the endocrine aspect, identifying early adrenal insufficiency, and then potential brain lesions and knowing what intervals at which we should be performing brain MRIs. And so this, I think, important pilot is really poised to roll out then across multiple Glia sites once he's completed it. Next slide. Just to mention that we are also clearly aiming here for improved understanding of therapeutics in the gene therapy arena. And at MGH, that has now become Mass General Brigham, we are piloting a first neurogenetics and gene therapy fellowship, which is GMA approved and allows us here to compare various gene therapy approaches and teach young investigators on how to execute on ex vivo lentiviral gene therapy, in vivo gene therapy, as well as ASO trials. And this is, again, knowledge that will be disseminated across our sites and across the young investigators who are maybe new to some of these modalities, making them more confident, making their staff and their coordinators more confident in engaging in these new modalities so that we can successfully execute on multi-center trials. It's a real medical education opportunity to teach residents in neurology who are coming out of residency and want to learn about gene therapy, how to manage these and execute these trials, also understand adverse events and how to interpret them, and really get clinical trial training that's so needed in the field of particularly child neurology. So we're very excited about this being a part of Glia CTN. Adeline?
Thank you, Florian. That was really wonderful. So, and so important and crucial so that one day Florian, Ali, and I can all retire and leave, pass the baton to someone else. Next slide, Omar. So I think, you know, our group thought a little bit as far as this particular audience, right? And thinking about how we could communicate to the disease community how important what each person's role in this effort can be. And, you know, you heard a little bit about how valued and relevant and important our patient advocacy groups are, and to what degree even they help drive our research questions. As Florian mentioned, the focus on the newborn screening effort for ALD came from our patient advocacy committee, as well as our other groups voting to sort of identify what the most important question was in the short term. But even beyond sort of the support that we benefit from the patient advocacy groups, it's also important to know that this is a partnership across levels, and, you know, even a partnership with individual families. So I'm going to talk a little bit about, you know, 4 different ways that even as an individual family affected by leukodystrophy, you can participate in clinical trial readiness for the leukodystrophies. And so number 1 is participating in a natural history study. So here I'm going to talk a little bit about the Glia CTN natural history studies, but what I'm about to say actually, you know, impacts also natural history studies that might be being conducted on the leukodystrophies that are outside of Glia CTN. Glia CTN does not do all of them, and there are natural history studies for vanishing white matter disease and Canavans and LBSL and others that are occurring outside of the specific projects in the Glia CTN. And so these are super important approaches that you can take on within your disease group that will really meaningfully impact how much we understand the disease that affects you or your family member, and how quickly we will progress towards clinical trials. Within the Glia CTN, right, you can participate in a natural history study, right, if you have any type of leukodystrophy. You don't have to have an in-person visit to a study site to participate. The study is open to all individuals all across the world, right? By participating, you'll be invited to provide a research blood sample for processing and storage in the biorepository, as I explained. You'll be asked to sign release forms and to give access to research for your medical records, because we learn an immense amount from medical records and from an understanding of natural history study. You know, if you are able, you might be asked to participate in outcome assessments that are performed by trained physical and occupational therapies, in some cases even remotely, because some of those are now, you know, we've learned a lot of lessons from COVID, and some of those are now happening remotely. And you might be asked to also submit online questionnaires or schedule a phone interview with a researcher. And these activities are all optional. You can enroll and then decide you don't want to do any of these things or do just one of them, right? And you can also change your mind. But if you're looking for a way to sort of give back to the disease community and try to help advance our understanding of these disorders and development of treatment, that's probably the single one most important thing that you can do. And you can, we'll leave this up for a minute, but you can go ahead and sign up to do that by participating online, right? And letting us know that you're interested. You know, our ethics boards make sure that you have to reach out to us and not the other way around, so that we know that this is something you want to do and that you do it of your free will. So if you put that into your browser, right, the glia.org referral survey, and I'm sure the ULF can share that with people if they need the information later. You will find a survey that asks you a couple of brief questions, basically allowing our research team to get in touch with you and direct you to the appropriate resources. Next slide. So the next way to participate is participate in a patient registry. Patient registries, next slide, tend to be run by specific disease groups, right? And specifically also by our funders at the NIH. They are providing support for contact registries for different disease groups that don't already have one. So, you know, if your disease already has a contact registry, like in MLD, like in from many ALD groups, that's wonderful, and we encourage you to participate. If your disease group does not, and you want to support your advocacy groups or support your disease community by creating a contact registry, there are resources available through the NIH that help support those type of activities. Next slide, please. And so the RDCRN, which is that large NIH group, right, has created resources that gathers and maintains information in a patient registry and facilitates, you know, information and recruitment for research, much like a different registry that might be done by an individual disease group. And, you know, this is done with all the sort of bells and whistles of security and safety that we can trust the NIH to do. Next slide. And so individuals who live with rare diseases, their family members and care providers or healthy volunteers can participate. And these groups can participate either individually, even if there isn't a specific registry to your leukodystrophy, or you can help us work with the NIH to create one that's specific to your disease group. And those are things for those groups that don't yet have contact registries that we're very willing to help support. Next slide. And so the RDCRN, which is the NIH contact registry, sees its partners as both the consortiums, the patient advocacy groups, as well as unaffiliated participants in ways just to create as much information as possible. Next slide. And so the NIH has created a very thoughtful approach, right, to allow participants that don't have a disease-specific registry to participate. Next slide, right? They also have made it easy for consortium and advocacy groups to create their own, either with sort of linking something that they already have that already is controlled by an ethics board, which some of our groups already have, or next, by creating new patient registries for groups that don't have the advantage of already having an ethics board and already having all the approval and the structure in place to permit them to do this. Next slide. And then they will also allow us to work with them to create additional registries and create mechanisms by which we can share information about our patients as well. Next slide. So thirdly, right, if you want to participate in information on building on the leukodystrophies, you can attend an event. And here are examples of events that have been led by the Glia CTN in the past, and we record those conferences, and you can check out our library of family conferences if you're interested in doing so, and especially with lifting of pandemic conditions, we hope to be doing additional conferences in the future, as is the ULF, I'm sure. And then finally, learn, next slide. You know, we have and are building more information about leukodystrophies at theglia.org. We already have some information about our sites and information about our partners, as well as information about specific data. And you could look, check back regularly, because we're adding more and more data about specific diseases, and specifically as we learn more and build more natural history study, we'll be putting more data there that you'll be able to visualize as well. All right. So, you know, lastly, we have about 10 minutes, right, to answer questions. And I think we'd be happy to take any questions as they come. My understanding is you're supposed to put them in the chat, right? I don't know, maybe we can ask the ULF if people are supposed to ask them out loud as well.
Yeah, that was an excellent presentation. Thank you. If anyone has any questions, they can type them in the chat, or they can use the raise hand option located under the reactions button at the bottom of the screen. But we do have a couple more minutes left.
I can only hope that the fact that there are no questions means that we've done such a good job explaining what we do, but I'm sure that there are still gaps or things we haven't thought about how we can better support the community. So feel free.
Can I just jump in and ask a quick question, if that's okay?
Yeah.
Really appreciate the presentation. Very helpful. So we're part of a new patient advocacy group called ADLD and the ADLD Center. Wondering how we can get involved. You described certain focus areas for you. We're not ready for clinical, as Dr. Eichler mentioned. How can we start getting involved in addition to the stuff we're doing around registry, natural history study? What else can we be doing right now?
Yeah, so I think it would be wonderful if you can reach out to Omar. Omar, maybe you can put the best contact information for our team, right? I'm sure you probably already know, but there are some efforts underway to work on early natural history for ADLD, and I think we can make sure that you're connected with those efforts and that we're thoughtful about how we can use the resources of the Glia CTN to support them. I would encourage you to also participate in our patient advocacy committee, so you hear about those resources that are available. You know, one of the advantages of that large Project One with the sort of larger natural history study is that we're enrolling not just diseases of focus, but all leukodystrophies, right? So we will, by definition, be sort of including patients who have disorders of interest to individual smaller groups. And I know we have a number of patients with ADLD in our existing data set. And so I think it'd be really just good to hear more about what your plans and goals are, where you are right now, and how we can make sure the infrastructure supports you. You also can work to have an investigator submit a pilot project, you know, for consideration of the committee and maybe to be sort of the seeds of a future natural history study. There's just a lot of different ways that we can try to be supportive.
Thank you.
I'll ask a question.
Oh, go ahead.
Oh, go ahead, Adeline.
Go ahead, Ron.
Yeah, I was just going to ask a follow-up to the previous question. And first of all, thank you guys for everything that you do. The reason that there weren't many questions because you guys are doing an amazing job. So thank you. But just as a follow-up to the previous question, specifically on goal number 1 around natural history, is there anything we as patient organizations can be doing kind of on a more macro level, you know, as opposed to individual patients' participation in natural history? Is there anything we can be doing to support, you know, progression of natural history work in our specific diseases, and including collaboration with your team together with other researchers outside of the network? Yeah,
I think, Ron, at some point, it's very much going to become an issue of, you know, we're building this infrastructure and this machine, and I'm talking for you specifically about 4-H, because I know that we've been collecting 4-H data, right? We're very much going to be collecting a sort of, or creating an infrastructure that basically almost in just the due course of clinical care collects a lot of natural history information, and it's going to become an issue of for specific diseases that really want to take that data, right, and analyze it in such a way that it's useful for clinical trials, creating a parallel infrastructure that can use that resource as a resource to then do the data analysis and do the data extraction that's needed, right? In the sense that we would almost need to have like 30 or 40 different fellows, right, attached to this program to work on each individual disease. If we were able to do each one of them, at some point it becomes easier to actually collect the data than it becomes to analyze and publish and disseminate the data, right? And so I think that as we build a richness of data, we'll have to think about how we resource ourselves as a community to do the data analysis. And it would be ideal from, Florian's perspective, if that was partnered with career development, right, for junior faculty. And so I think there are lots of things we can think about, but that's going to end up being at some point, we're building a machine, but then we need to have the people to sort of take things out of that machine and package it up in ways that are useful, right? Because some of that doesn't happen in an entirely automatic way. We're trying to build some automatisms into it, but it will never be complete. Does that help?
Yeah, that's amazing. That's really helpful. And that's a sort of a project I think that a Bayesian organization can bite off and chew, as opposed to the machinery, which is much bigger. So that's great. Yeah, thank you.
Yeah. And that's where we're looking for a partnership. And then somebody asked a question, if we have participated in meetings and given blood and you have seen us in person, and by that I mean people are directing that at me. I don't know who is the person who's addressing that question specifically. So if you've been enrolled in a research protocol at either Children's National and you consented to that data being moved, and or you've been enrolled at Children's Hospital of Philadelphia or one of the Glia CTN affiliate sites, you should most likely expect that your data is being included, and you can always reach out to us via email at theglia.org. I hope that also, Inderpal, you saw that email, so you know how to get in touch with us to verify that that's the case and that no updated consents are needed. Thank you for asking that. Okay, I know we're at time, and I know we're gonna get, Bob. Perfect. Thank you, Bob, for that comment for Inderpal. Okay, I think we're going to get shut down automatically at 2 or close thereof. And so I want to thank everybody who participated in this session. I want to in particular thank Omar and Florian and Ali, who are our partners and collaborators in all these efforts, and look forward to continued work together as a team. Thank you very much.
Thank you. Thank you to our speakers, our sponsors, our audience for making the virtual family conference this year a possibility. We really hope to see you in person next year. Thank you.
I hope so too. Bye.
Thanks, everyone.